Hey all, welcome to ShareTrek. This is Raj here. Friends, today I want to add to our understanding of the ever-evolving gene editing technologies field from an investor perspective. By saying an investor perspective, what I mean is that we will try to understand at a high level what these new developments are, and I will drill down a little bit where needed into the actual technology so that our understanding can be comprehensive. The objective is to improve our vocabulary and awareness of the gene editing technology so that we have it in play as we evaluate the evolving valuation of various genomic companies in our watch list. And if we did not have this tracking of the latest developments, then the chances are that our valuation of the companies might not be accurate. So with that said, today I'm going to talk about some advances in prime editing, and this comes from an article in a website called phys.org, phys.org. So let's get started. Welcome back, friends. Let me show you the article that is the source of this segment and then proceed to provide you with a summary of what this article tries to convey. This article discusses recent advancements in gene editing technology, also known as prime editing, particularly the developments of improved versions of this tool by researchers at the Broad Institute of MIT and Harvard. Here's a summary of the key points. The first bucket of finding would be prime editing technology. Prime editing is a precise gene editing technology introduced in 2019 by David Liu and his lab. It allows scientists to make targeted insertions or deletions and other changes to DNA without breaking the double-stranded DNA. And this has its own advantages, which we have spoken before, so I won't take too much time on it. Suffice to say that prime is an enhanced and more precise gene editing tool. The next point I would like to summarize here is about the enhanced editing tools. Researchers at the Broad Institute have used continuous laboratory evolution and engineering methods to enhance prime editing technology. Their new editors are more efficient and specialized than previous versions, and they seem to have 7 such modified tools, each for a specific application. We'll talk more about the general characteristics of all these tools in this video. The next aspect is wider applicability. The improved prime editors are capable of modifying DNA in cultured cells and animals that were previously challenging to edit, including immune system cells and brain cells. The broader applicability opens up new possibilities for genetic disease treatment. The next bucket I would call as smaller editing molecules. The editing molecules used in these advanced prime editors are smaller, which may make it easier to deliver them into the cells and into various parts of the body for potential therapeutic applications. And friends, if you recollect, when we started our coverage of CRISPR-Cas9, one of the main disadvantage of CRISPR-Cas9 was that it was a bulky molecule and transport mechanisms were going to be a challenge. But prime editing and the latest version of the prime editing tools overcome that challenge altogether. The next aspect I would like to summarize from this article is that prime editing involves a combination of molecular machines, including a disabled Cas9 protein, an engineered prime editing guide RNA or PEG RNA or PEG RNA, and an engineered version of an enzyme called reverse transcriptase. These components work together to make specific changes to DNA. The focus of this research was on enhancing the reverse transcriptase component of prime editing, which has not been previously optimized. Using a directed evolution approach called PACE, the researchers generated new reverse transcriptases that significantly improved editing efficiency. Let me take a brief segue from here to explain a little about the reverse transcriptase. The reverse transcriptase in prime editing is responsible for copying the RNA template in the PEG RNA into a complementary DNA strand, allowing for the precise modification of the target DNA sequence as specified by the engineered PEG RNA. The enzyme, this particular enzyme, is a critical component of the prime editing system, enabling the creation of specific and accurate DNA edits. Next, I would like to talk about the specialization of these editors. The evolved prime editors were found to specialize in specific types of edits. The researchers have now identified rules for when to use each of these 7 prime editing systems they developed, and those guidelines can help researchers pick the right tool for the right applications, and thus enhance the efficiency of the projects. Next, I would talk about the therapeutic potentials. The new variants of prime editors demonstrated the ability to insert longer stretches of DNA in human cell lines and the brains of live mice, greatly increasing editing efficiency. This could have significant implications for therapeutic applications in treating genetic diseases. Finally, one of the aspects is the compactness of the molecules and therefore the impact on delivery. These new prime editors are more compact than previous versions, which makes them easier to deliver into animals and potentially humans, improving their practicality for therapeutic purposes. And in terms of future applications, the researchers are optimistic about the adoption of these advanced prime editing tools in the field of gene editing, and hope they will contribute to further progress in the life sciences and medicine sector. Overall, this research represents a significant step forward in the development of prime editing technology, with the potential to revolutionize genetic disease treatment and other gene editing applications. Friends, let me also add that I'm very optimistic that these prime editing technologies can be very useful for development of new HIV therapies, where prime editing components can be used to directly target HIV, which are lying dormant in macrophages, dendritic cells, and CD4 T cells, and neutralize them in some way or the other. Well, friends, that brings me to an end of this segment on prime editing enhancements. It seems like prime editing is going to be the future. And when we had CRISPR-Cas9 discovery, there was a lot of excitement, despite the fact that CRISPR-Cas9 had its own limitations and the difficulties in doing double-strand breaks and off-base edit possibilities and all that. But with prime editing, things have got better, and now with these enhancements we have got even more precise tools for each application of editing. So this places more and more potent and precise tools in the hands of researchers. Therefore, this is going to contribute to better treatment of various genetic diseases, and also better editing of potentially harmful viruses in the body, so that they can be disabled. And I suspect HIV will be one of those viruses, and we may hear something about that in the near future. So with that optimistic note, I would like to conclude this video. And also, friends, please do not forget that the researchers who are doing all these research and coming up with all these refinements are also behind prime medicine. So I think that we need to take a second look at prime medicine at the earliest opportunity to see what their prospects are. So with that said, I would like to bring this video to an end. Thanks and have a great day. Bye for now.